Hello, my name is Peter Bandettini, and I'd like to thank you for inviting me, specifically Alan Wagoner, um, to give this pre-recorded lecture. Look forward to talking about this in a few days. Just to begin, the the main thing I really want to talk about today is high field imaging and layer fMRI. And I know that Renzo Huber previously gave a talk, and this talk will overlap slightly with some of what he had to say, but definitely be very complementary in many ways, since I have my own take on layer work that we've been doing in our group and in collaboration with Renzo. So the main overriding theme of my talk is pretty much deriving more information from fMRI data, from layers to dynamics. And so I believe even more firmly now that there's more information in the functional MRI data, the time series, the spatial information, than we've been able to extract so far. And so there's a lot more to be done, and this is one direction. So the goal of all of my research, pretty much at the NIH, is to develop and refine methodology for extracting information from the MRI signal. And that includes all of MRI, but mostly functional MRI. And that's really towards two goals. One is just understanding the brain. How can we use fMRI to better understand the brain with all its limitations in time and space and interpretation? And the second goal is to really bring fMRI to the clinic, to be able to have fMRI as a useful clinical tool, which I believe it can be. I won't really be talking too much about that today, but I just want to give that overview. And the way I see it, I think that there's actually four pillars of fMRI research. One is the MRI pulse sequences and hardware development, signal processing methodology, interpretation of contrast mechanisms in physiology, and basic research, as well as clinical applications. And these are all very complementary. And an advancement in one helps the others advance as well. And so they all feed off each other as well. And a lot of the, I believe, better application research sort of is at the interface of all of these. They use the most advanced hardware, they use the best signal processing methods that's tied in with how they understand the signal. And I believe what I'm trying to do is position my group, the section on functional imaging methods, right at the interface of these, mostly not really doing a lot of actual applications, but sometimes we do as proofs of concept, but mostly at the interface of signal processing and understanding contrast mechanisms. So this is a busy slide. It's just to give a perspective of over the years, at least since 1999, when I came here to the NIH, we've been really busy in really pushing many different avenues of understanding the signal, developing pulse sequences to be more specific to specific types of neurovascular coupling, developing signal processing methodology, everything from multivariate mapping to talking about global signal regression and various other techniques for extracting time series data. And also interpretation and physiology, just doing various stresses to selectively cause blood flow changes without activation, looking at latencies and widths and things like that. So after 30 years, that was only like 20, but after 30 years of fMRI being around, where's the room for improvement? And I'd like to give this overview slide because it just really helps put things in context. One, we're still working on fully separating the informative signal from the noise in fMRI time series. There's still much to do in terms of modeling the noise and modeling the signal so we can extract them. And as most of you know, the controversies of like whether should we do global regression or not, or things like that, all have to do with what we consider noise and what we consider useful information, or information that we really don't want to throw out. So the second is understanding the neurophysiological relevance of all fMRI measures. So right now, over the past, at least 20 or 30 years, we've been mostly looking at the magnitude as sort of a ad hoc measure of the degree of neural activation. But there's all kinds of other things in the signal. There's transients, there's spatial correlations, there's undershoots, there's latency differences. All that can be extracted and used. We also want to make sense of the fMRI data. So we have all this data, and what we typically do is map it. And realistically, just mapping it and looking at the data is great for papers, but it doesn't really get us anywhere. Just simply showing where something happens or when it happens is kind of like cartography. It doesn't give us an understanding of how the brain is organized beyond the superficial spatial locations, which is probably very important, but it isn't really getting into the computations of the brain. And I don't even know if fMRI can get there, but the one area that I think is fruitful is working on trying to get a hold of this data and apply it to network and computational models. So integrate those models with the data to sort of derive new insights. I think we have a long way to go in terms of increasing our spatial and temporal resolution. The better we understand the hemodynamic response, which is the limiting factor in both, the better, the higher resolution we can go. And I believe we can probably go down to less than 0.5 millimeters, certainly in space and in time. There's been some, preliminary data that suggests that the hemodynamic response is well behaved as long as we understand that its latency varies over space due to how you capture the vasculature. This probably can be pushed down to on the order of 100 milliseconds. So that's one thing that can be improved. Integrating information from multiple modalities. So certainly simultaneous EEG is extremely complementary to fMRI data. But it doesn't necessarily even have to be obtained simultaneously. You can get MEG data, you can get PET data, all could tie in to better understanding the fMRI signal. And of course, there's been recently reproducibility issues. I wouldn't say really a crisis, but more we're coming to the realization that we need to be more reproducible. So, and this actually not only involves best practices and reporting all the metrics as we pool our data, but it's really understanding and capturing individual variation that contributes so much to variation. So it's both the methods and understanding really what we're looking at, and not really expecting people to always be the same, even in selected, somewhat homogeneous populations. And then finally, clinical applications. The holy grail of fMRI is to develop biomarkers for psychiatric disorders, to be able to say, oh, this person will respond well to this sort of treatment or drug, or this person has this type of schizophrenia. It would also be nice to be able to put individual subjects in the scanner, and as I was implying before, to be able to put a subject in and say something about their functional activity as it relates to their behavior in a robust manner. That's sort of the bottom line for clinical application. You can't use group data. And having real-time fMRI for neurofeedback and things like that, I think is also very promising. So let's just go into the brain a little bit and where fMRI plays a role here. So the brain is organized across many temporal and spatial scales. This happens to be the spatial scale. And to truly understand the brain, I believe you have to really understand all these scales. Unfortunately, fMRI is limited to this scale. And with layer fMRI, we're sort of knocking on the door of looking at circuits. And this is just reiterating what I said before, that there are many issues in terms of trying to derive neuronal information, everything from inhibition, excitation, different frequencies, transient, spontaneous activity, through this hemodynamic transfer function, which actually varies considerably in space in sort of a semi-random way, depending on how the vasculature is captured, to the measured signal. And the measured signal can be magnitudes, latencies, correlations, fluctuations, transients, undershoots, and so on. And also I've added layer position as well. That is an informative aspect of the measured signal that will inform directionality, which I'll get into. So, and of course we have to deal with noise and better understand it and model it. Thermal system motion, physiologic cardiac respiration. And a lot of this might be mixed with the neural signal in time and space. So it's truly challenging, and we haven't yet completely solved that. So this just shows that if you divide the brain into voxels, it certainly can capture large vasculature, but this problem even becomes greater when interpreting layer fMRI data, because you typically have arteries that feed into the layers that are larger. You have descending arteries and ascending veins that then spread out the hemodynamic response. But if you're really relying on differentiating between layers, you really have to either find a way to calibrate to these larger vessels that smear everything, or develop pulse sequences that are sensitive just to capillary effects, which are somewhat, but not perfectly, more homogeneously distributed across the cortex. So that's the challenge there. So definitely having big vessels is a problem in interpreting very low resolution data, but a little bit less of a major issue than it is at the level of layers. So Renzo showed this diagram. It's useful to sort of get a gestalt of kind of where layer fMRI fits in. We can measure the brain and what it relates to across many different spatial scales. Most of fMRI has been limited to within area sort of maps to large blobby brain areas, which is fortunate that the brain is organized at that scale in that way, since we can actually pull it out and trying to tie this in with behavior. But looking at layers as well as columns helps to potentially unlock the intermediate circuitry that connects these two. One diagram that he didn't show that I'd like to show is that all these other high resolution techniques are very limited in their spatial coverage. And so you have to make very, very specific hypotheses as to where the activity is. And you can't say anything as to how they interact with the rest of the brain as easily as if you were to collect whole brain data. And now we're able to collect whole brain data, so we can actually look at layer and column information pretty well at the whole brain level. So that is nice in the sense that we can go to this fine resolution that bridges the gap between these methods and look at it at whole brain. So why is layer fMRI kind of important? And I believe that it is because at least emerging models of the brain suggest that it's essentially a prediction machine. And how does it do this? Well, there might be many, many different levels in a hierarchy in which in the cortex, sensory input is compared with a prediction, some sort of model of what's expected. And this can be very hierarchically driven all the way from low-level sensory perception to much higher level sort of conceptual prediction and receiving of predicted behavior or whatever. So this may be very hierarchical. The idea is that these areas in the hierarchy where these sort of computations are taking place, which we're still not clear on what exactly they are, occur in the cortex across layers. And there might be layer modulation depending on whether there's feedback or feed forward information. So this is the basic, super simplified diagram of what our hope is with layer fMRI. That, for instance, the idea is that, like for instance, if you have V1 speaking to higher brain areas, that will be feed forward information that usually terminates in layer 4 or somewhere in the middle. And the higher brain areas usually when they feed back information to V1 or other visual areas, it's typically across the superficial and deep layers. So, and also what's interesting, which is still not completely established, but it seems that these have very specific frequency contents, whereas gamma frequencies might be the feed forward, alpha and beta frequencies might be feedback. And this can be shown as a sort of like, and I'll be showing more and more of these plots in my talk, these canonical sort of single hump, which indicates a feed forward dominated column. So some area that's higher than the area being that it's talking to, that's receiving input from. And this is feedback dominated column. Let's say this is in V1 receiving input from a higher area. It would show like this double activation here, or double hump right here. So what do you need for layer specific fMRI? Well, you need small voxels, and you can do it with less than 1 millimeter. The smaller, the better. We can now at 0.75 or even down to 0.4, I think we've gone. You can resolve maybe 3 levels. I wouldn't say layers because there's at least 6. So you can't resolve the layers themselves, but you can kind of get a rough approximation of where the activation occurred, either upper, middle, or lower. You need very, very high sensitivity. Of course, the signal noise is directly proportional to the voxel volume. And after averaging, you typically are working with a signal to noise of about 20 to 1, which will give you enough signal in the amount of time that you have to image. And this is achieved at high field, also with local RF coil arrays, which are very important as far as this is concerned. You need high, and also which you can also do many different ways of regressing out the noise if you know that it's noise. High specificity. There have been a lot of attempts with various pulse sequences. Spin echo is great, but as spin echo echoplanar has a wide readout window that's highly T2 star weighted, so it's not really a pure spin echo. It also suffers from a lot of intravascular signal, even at 7T from the red blood cells themselves. So it's not quite as good as it was hoped. Arterial spin labeling is great in terms of specificity. You're getting at perfusion. However, its sensitivity is so far way too low to practically image activation at that resolution. Vaso technique developed in the early 2000s, but now has become really useful because it turns out, and this wasn't really expected. I thought the typical Buxton balloon model suggests that big vessels sort of expand with blood volume changes, but it turns out it seems that the blood volume changes occur at the microvessel level selectively. So that's actually surprising, and really, really good news for vasocontrast, since it seems to be more localized. Vaso suffers from low speed and narrow coverage, where Renzo has been able to increase the coverage of vaso. But it's still kind of slow, and so you don't quite get as much efficiency. 3D grays is a technique that has been developed that seems to work pretty well for being selective as well. It's a combination of spin echo, but it's done in a slightly different way that's less sensitive to these large vessel effects. So I'm not going to get into that sequence, but it's a sequence that seems to work. And of course, you need novel processing. And Renzo talked a lot about this in his talk. You can't just do your standard way of processing. You'll miss all the interesting information. You have to develop and do the processing with the idea that you're going to be looking along cortical ribbons and tangentially through them. So that's involved. So this is a classic plot, but I always like to emphasize it for people to get an intuitive knowledge of what's going on. So typically with fMRI, we consider temporal signal to noise. We never really consider image signal to noise, because you could have an image signal to noise that's 5,000 to 1. but you really are limited then by the physiologic noise, which gives you a ceiling of about 100 to 1. Until we can completely filter out all physiologic noise without getting rid of neural activation, we're stuck with that upper signal to noise. And I think we can filter it out. So then we could really increase our sensitivity. But when we're at the resolutions we're trying to go for, on the order of 1 millimeter or less, going to 7T is critical. I mean, already you're very much in what's called the thermal noise regime, where this ideal line of where temporal signal to noise matches image signal to noise is close to what you're actually doing. Physiologic noise is a factor, but not a big factor. So as you go to even higher resolution, your temporal signal noise does get down. Just calculating it just based on the voxel volume. And at 3 Tesla, this is prohibitively low. Although due to, once again, Renzo's work, there's some hope for doing this at 3 Tesla. 3 Tesla does have some advantages, that the T1 differences between blood and tissue are larger, so it enhances vasocontrast. And there's also, improved image quality at lower field strength. So just in a nutshell, the advantages: higher signal noise, higher susceptibility contrast. This also helps fMRI bold, but not only that, it also helps structural imaging. And the higher signal noise, less than 1 millimeter, this makes all the difference. It's the difference between scanning maybe 50 minutes to get enough signal to see something versus, 5 days to see something. That difference in signal to noise. The challenges, of course, at 7 Tesla, there's more RF heating, there's RF spatial inhomogeneity, there's more, magnetic field inhomogeneities that cause more signal dropout. T2s and T2 stars are much shorter. The T1s are longer, so you have to wait longer for the longitudinal recovery to happen to have enough signal. And also, as I mentioned, there's smaller differences between T1 and of tissue and blood, so that reduces your contrast that you have to work with. So Renzo showed this. I like to illustrate this to say, hey, there's a lot of scanners. That's certainly true. I think there's up to 100 now. This is a slightly old version of the slide. But one thing I like to show this slide, but I like to sort of mention that, it's amazing that vendors are taking this risk to develop 7 Tesla. But I don't think that 7 Tesla is sustainable on just 100 scanners and just for research. There really has to be more clinical applications, like real clinical applications, to justify the cost of 7 Tesla. And I think they're playing this out and waiting to see what happens. And hopefully the clinical market opens up. And so, certainly if fMRI becomes clinically relevant at 7 Tesla, that will open it up. But I think in the meantime, I think every group using 7 Tesla, if it's primarily for fMRI, they should also be thinking of other sort of structural applications at 7 Tesla. So on our scanners, since we had a 7 Tesla from, we had one that's longer, but this is from 2014 to 19, most of our applications on our 7 Tesla have been structural applications and not fMRI. We've done fMRI, we've done methods development, we've done just a little bit of spectroscopy. And here's an example of a clinical application. This is something that cannot be seen at 3 Tesla. So this is work by Danny Reich's group in NINDS, basically showing that they can identify and differentiate MS based on the location of the enhancements in MS around vessels. It's typically associated with vessels, and you can see the vessels because of their susceptibility effect. This differentiates other types of dysfunctions that cause white matter inflammation that are not MS, that look a lot like MS. And at 3 Tesla, you can't differentiate this as well, and nearly as well. Here, you can do it 100%. So there's a clear clinical application based on the susceptibility contrast. But I'm not going to get into that. I'm going to mostly get into layer fMRI from now on. And once again, I just have to reemphasize, Renzo has been the driving force in my mind of most of layer fMRI in the world. And he's extremely humble, but he should be given appropriate credit for all the work he's doing driving the field. And also, if you check out his website, it is the clearinghouse of everything layer fMRI, and it's awesome, and it's really super helpful, and he's super helpful. So, and he's also super knowledgeable and smart. So it's been great working with him, and we're lucky to actually be getting him back at the NIH this fall to start out as a staff scientist in our group. All right, these are all of our collaborations that are related to the work I'll be showing. So I'm going to go over this pretty quickly. This is the sequence, the sort of the bread and butter sequence that we have been using. It's a simultaneous vaso and bold data collection where you give an inversion pulse, you wait until the blood goes through an endpoint, and then you image to get the vaso, but then you wait again till they're both above the null point to get the bold effects. And the idea here is that with vaso, you can actually differentiate layer activity, whereas bold, it's typically weighted towards these peel vessels right here. And one additional bit of processing that we can do because we've collected this bold and vaso simultaneously is that we can actually even further clean up the vaso signal by taking the two time series, averaging them, and then dividing them to get rid of any susceptibility effect, which should be identical for both of them, to just leave the vaso effect. So that further cleans things up. And this just shows you sort of graphically, what improvements really mean at this higher resolution of 0.8 millimeters. You go from a blob of activation to specific layer activation. And there it is right there. So I'm going to talk in my remaining 25 or so minutes, an overview of some of the applications and some of the work that we've done from my own perspective. So, certainly we're trying to calculate what's the best pulse sequence to use. And this is work that Renzo's done early on. And it's funny, it's early on, but it's published in 2021, but it was around a little bit before, in collaboration with the Minnesota group as well, basically showing that if you track sensitivity versus specificity, and he had specific measures of how sharp these profile curves were, and then how high amplitude they were, you get bold, which is great in terms of sensitivity, very non-specific. And they all seem to fall upon this sort of curve. This is T2, a pure T2 sequence with diffusion weighting to get rid of the intravascular signal. You have nothing left. But somehow, which we're still getting our heads around understanding, vaso jumps out of this curve and gives you the best trade-off in sensitivity, enough sensitivity and enough specificity to actually be practically useful. And here, the corresponding maps of the various techniques. And vaso does seem to look like the sharpest and sort of the cleanest of all of them. What's interesting, though, and I think this is an important point, is that vaso is great for layer mapping. It seems that if you map the layer profile, you see vaso is very sharp, whereas bold is sort of this general curve, and sometimes it even is way higher at the CSF level with peel vessels. However, and that's perfect for layers. However, if you look at the columnar profile, and this is basically a plot of the ribbon along the visual cortex, and you find that all the areas that show the susceptibility effects, these are the large vessel effects, are also somewhat showing up in both bold and vaso. So it means in some way that that there's less spillage of the vasculature along the cortical ribbon. So if you're doing things like retinotopy, and you want to do retinotopy really high resolution, you might be just as fine doing bold, if not better, because you have more sensitivity and there's no spillage along the cortical ribbon. Whereas, because it seems like the vessels get the blood, it comes up, and it might spill along the peel vessels a little bit, but less than we expected. And this has direct effects. This is some work, preliminary data from Eli Merriam and IMH, basically saying that bold seems better for detailed general activation discrimination. I'll bring up one exception to that with our digit mapping, but VASO is better for layer-specific discrimination. So laminar mapping. So this was Renzo's first work that made an impact. It was published in Neuron 2017. I'm not going to go into too much detail, but basically it's a proof of concept that if you look at the motor cortex, the theoretical model is that cortex and thalamic input occur. It's organized slightly different than what I was talking about before with other layers, visual cortex. Its upper layers are thalamic input, lower layers are spinal output and thalamic output. And so we had various tasks where we tapped with touch, so that had sensory and motor, and you had a double hump in motor M1. If we tapped without touch, you have much less sensory, cortical sensory input, but similar amounts of output, but maybe a little bit less because you're not quite moving as much. And if you touched only and had input but no output, this is the black curve. You have a little bit of sensory right here, but absolutely no, if anything, is inhibitory of the output. And then finally, if you did the ipsilateral side, it seems to inhibit the sensory inputs or the cortical input and very little output. And bold effects, you can kind of see this a little bit, but clearly it's weighted by the CSF effect. So really nice, clean results that help push the applications in the field. We also can look at resting states. So this is just, once again, sort of a proof of concept study where we took that same data and we just took seed voxel approaches. We used looked at the upper and lower level with the seeds, and we saw what was correlated. So if you look at seeds in superficial layers, this sort of light blue, you see many more areas of activation in the sensory cortex, which is as expected. If you took the seed in the green upper or lower deeper layer, you have a lot more spontaneous activation that occurs in premotor cortex, which suggests that this is feeding into sort of feeding into premotor cortex. And of course, it would be great to look at the layer profiles there, but we didn't do that. But so if we just flip this around and then take big seeds, a big seed in S1, a big seed in premotor, you see the same sort of things. A big seed in S1, it feeds into the upper. If you use that seed, see how it correlates across the same M1 profile. It's a large hump towards the CSF and a double peak when you take the premotor seed in the M1, which suggests that M1 is lower in hierarchy than premotor, which is feeding back this information to motor. Whereas here it's higher than sensory. Sensory feeds into M1, I guess maybe for proprioception, other things. So, and that's higher. So it's feeding into M1, it's feeding back, getting feedback from the premotor. So as I mentioned, we're not only doing layer mapping. This is a really nice experiment that Renzo did in which he just thought, well, let's see how far we can push the resolution just with looking at columns or in individual digit mapping. So many people have done this and they get something like this. Whereas in sensory cortex, you get some differentiation. Motor cortex, it's a little messy, but you get some differentiation of each digit. Looks pretty good. But with VASO, it just pops out. And there's some interesting information that pops out here in that with VASO, the sensory cortex seems to be organized with one pattern that goes from the index finger down to the pinky finger in the sensory cortex. In the motor cortex, it seems to show kind of similar in some ways to V1 and V2. There's sort of a mirror image pattern that seems to emerge where the pinky finger shows activation, it goes down to the index finger, and then it flips an index finger back to the pinky finger. So, and this showed a really nice retest, repeatability. And as we thought about this, we thought, okay, let's try a slightly different task and see how that maps. So we had a macroscopic task. In this case, it was doing extension against the rubber band versus flexion against a ball. And what we found there is that with the extension, there's this very specific area activated. And with the flexion, mostly that activated sensory cortex, since most of your sensors are in the inside of your fingers, I guess, and some other areas as well here. And that just shows the time courses of those. So there was still activation here for the flexion extension, but not as much as with flexion. So, and we looked at the pattern of that activation of that macroscopic task and compared it with the mirror image pattern in the finger tapping task. And we find that they really do align. Essentially, if you line them up, you can see that this mirror image is organized exactly around the macroscopic motor motion. So what we found is that, one, we found that there's a mirror image pattern, and two, it organizes itself across so digits, the activation of digits when you're extending your hand versus flexing it is very different. And that's how the motor cortex organizes itself across digits. So that was, I think, a brand new finding with this technique. So there's other approaches too. There are, ways of looking at the digit representation just using resting states. And this is a very kind of crude but intuitively graspable approach. We basically did ICA of the whole brain. We zoomed into a specific network of the ICA and did ICA again. And we found these subcomponents of this network. And then we zoomed in to these two, did it again. And finally, after we zoomed into this one, we found that it differentiated itself into digit formation. So this is sort of like an iterative ICA approach, probably very similar to using partial correlations or other approaches, but this works really well and it's sort of intuitively graspable. So we've took it a little bit further with a visiting scientist in CalU in our group, and we just extended it to sensory. And we thought this is sort of more towards the prediction engine sort of concept, in which we looked at sensory cortex. And first, as a proof of concept, we localized the sensory cortex, and then we specifically modulated, activated specific digits. And the hypothesis here is that with the finger sensory input, it goes to the thalamus and it terminates in layer 4. And what we found is that if we stimulated D1, it activated layer 4. If we stimulated D3, it activated layer 4 for D3 and localized right here. So that works as far as sensory input. And we took this a little bit further and made a task in which we either sequentially stimulated the fingers or randomly stimulated the fingers. And one involves prediction. So you predict, you expect the index finger to be stimulated. Here, you don't expect anything. You don't know what to expect. It's random. So when you expect the index finger to be stimulated, it turns out that in D1, it shows upper, middle, and lower activation when it's actually stimulated and you're expected to be stimulated. If you're doing it randomly, then you still get the input from the thalamus, but you get no upper and lower cortex activation. So there's no top-down sort of expectation signal. If you do an expected signal in which you don't stimulate the finger itself, you get lack of activation of the thalamic input, but you do get some additional activation in the upper layer, at least, with the expectation response. And if you don't stimulate this finger at all and then keep the random sequence, the other 3 fingers, you get nothing in D1. So that's actually was a really nice demonstration of the expectation signal modulating upper and lower layers independent of the middle layer activation. So we also extended this to more of a cognitive task. Emily Finn, among all the other things she's been doing, she collaborated with Renzo on this particular project, and she wanted to look at working memory. And so the idea is that, once again, when you have fMRI, you just see these blobs of activation, and you're not sure what does what exactly. And while we didn't necessarily answer the question, since we didn't look at the rest of the brain, we only focused on dorsolateral prefrontal cortex, we did at least start to make headway into understanding at least what dorsolateral prefrontal cortex is doing at a layer level. So the two tasks were subjects would see letters, and they were asked to either remember or alphabetize them for a certain delay period. The alphabetization was much harder. And so you would expect in this model, with these recurrent connections in the upper layers, more activation, whereas not much modulation in the lower layers. But then when they were asked to respond or not respond, this lower level that speaks more to the premotor areas would be activated. And it turns out that's what occurred. With the maintain and manipulate, you had a modulation of the upper layer activity. And with either having the subject respond or not respond, I mean, they did the task and they would say respond or don't respond. If they responded, you had a clear activation in the lower level of lower layer of dorsolateral prefrontal cortex, but no activation at all when they didn't respond. And so this was clearly differentiated here. And in this case, actually, it seems that the prefrontal cortex, dorsolateral prefrontal cortex cortical thickness is thick enough that you could seem to differentiate it pretty well with BOLD and VASO. VASO did a little bit better job, but BOLD did a pretty good job as well. So there's some hope in maybe using BOLD for prefrontal cortex layer work. So that was very nice, very clean. Didn't answer the whole question of what the whole brain is doing in terms of manipulating and maintaining working memory, but it helped at least show that there's layer activation. So that's the summary of that. Resting state connectivity for mapping feed-forward feedback connections. So this is a nice study where we first localize specific areas. The lateral geniculate lucus, which is the lowest level in the hierarchy, it receives sensory input, goes to V1. V1 was selected based on where the stria generi were. So that was the boundary of V1. And we also looked at MT based on just simply a localization task that involved moving dots versus static. So clearly localized MT. And so our first step in doing this was looking at a V1 layer profile. So we took the cortical ribbon in V1, and that was what we looked at. And then we picked seeds from these other areas. So first, if we picked a seed in lateral geniculate nucleus, which feeds into V1, you see a resting state correlation that peaked in the middle layers. So that's as expected. So you could discern the hierarchy that this was lower than V1 based on picking a seed here and seeing that there's a hump here. If we picked a seed in MT, which is higher than V1, you could see that in V1, the layer profile, this is looking at the same signal, but just how well it correlates with the seed. It correlates much better with the seed in MT in this double hump manner. So that was great. Very clean, very nice demonstration of these two kind of canonical functions that we're seeing again and again and again. So we took this one step further and made templates and allowed the seed to just travel. So we took a template of either receiving input from lower levels or receiving feedback from higher areas. And we started out with a seed in the thalamus. And so the whole visual cortex relative to the thalamus had this single hump. So there was a middle layer input from the thalamus. And as we move the seed along the visual cortex, what's interesting is that all the areas that were in the more, starting out more medial in the visual cortex before we moved out in the cortical ribbon, they all showed relative to that seed location, this double hump. So this area sort of speaking to these areas it's feeding back to those areas. As we move the seed along, you can see that all the areas beyond the seed are blue, which is the seed is still speaking to those. It's lower than those, but it's feeding back to all these other areas in this manner. And we take it all the way up to MT, and it follows that pattern, which is very, very, very clean pattern as far as that's concerned. And we even made a movie of this. So if you move the seed along, the entire cortex starts to turn, the entire visual cortex starts to turn blue as you move the seed up, because now this seed is feeding back to all the other areas. I'm sorry, it's feeding back to all the other areas here, but still MT, it's still feeding forward to MT, and MT has this profile here. So this is a potentially very, very powerful way of looking at cortical hierarchy just using resting state. So we've also, preliminary data, we've seen that if we look at just simply looking at the global power, the power of global signals in all of cortex, we see that it seems to reside mostly in middle layers. So maybe it's receiving some input, maybe from the thalamus, whatever, that's causing these global fluctuations. So we're just looking at the global correlations here and how much they, what the layer profile is. So that's kind of maybe a hint as to what global signal might be. Whole brain at high temporal resolution vaso. So Renzo has developed methods that take us beyond just simply having these little slivers of cortex into whole brain, using, not relying on the fact that you don't necessarily need to know the blood, you just need to set up a difference in the signal between blood and tissue to actually see this. It's a little bit lower signal to noise, but it works really well for whole brain. I have to mention also, Yuhui Chai has developed a new tool called Vapor. Slightly different contrast mechanism that I won't get into, but it also works at whole brain, and it's very sensitive. It's a combined vaso and perfusion that helps boost the signal a little bit without sacrificing specificity. So, and this is just, what the whole brain images look like. It's really nice. You can see layer activity in the whole brain. But now the problem is we have this explosion of data. So here's basically a massive correlation matrix where we just divide the brain into cortical areas and see what cortical areas correlate with other cortical areas. But now we've just exploded this, that each cortical area has a layer profile. And so now you have this squared again in terms of what you can look at. And so, Renzo talked a little bit about this diagram where you can actually make very specific questions about very specific networks and see how they interact. I really think that the other method of looking at seeds and trying to then identify areas that you then see how the seed correlates with those cortical layers is really powerful and is very hypothesis driven in terms of pulling out feed forward versus feedback in areas that are connected. So here's one preliminary data from a student of mine, Richard Klein, in which he looked at V4, which is positioned sort of between, it's higher than the other visual areas, but lower than cortical areas. And he's looking at the layer profile. These are all layer profiles in V4 from the various seeds that are listed here. So if you look at BA8, BA9, BA10, these are all upper cortical areas. They seem to show this double hump in V4, which is the feedback that occurs. So these are based on this double hump. They're higher and they're feeding back to V4. If you look at the areas that are potentially below V4, LGN, V1, V2, V3, it's all this. And once again, we're all looking in V4 layer profile. You show this resting state correlation that's a single hump. So it's a nice way of pulling out upper and lower cortical hierarchy. And lastly, you can actually sacrifice some temporal resolution or some spatial coverage to increase your temporal resolution of vaso to start to look at these transients. Just here's this one example of where he's using magic vaso, which is the rapid technique at a very limited area, single slice, to pull out the dynamics of blood volume return to baseline. And it shows a clearly different time course than BOLD, which is interesting in itself. So now finally, future challenges. So I think that one, we're nowhere close, but we're getting there towards standardized processing. We're even still trying to figure out what a standardized processing pipeline for layer fMRI would look like. Whole brain connectivity and hierarchy. I think there's a lot to be done with maybe some of the tools that I mentioned. We need, if we're going to use BOLD, which in many cases we might just have to, there are calibration methods that could be useful based on either breath hold or some sort of modeling of the signal fall off from upper to lower layers. How do we multi-subject average and compare multiple subjects? So spatial registration is nearly impossible at the layer level unless you do some extremely nonlinear work or very focused on specific cortical areas. So it's really hard you might need something like hyperalignment potentially. How do you do multi-subject averaging in comparison with layer fMRI? That's an open question. Incorporation of layer activity with computational models, certainly since we're getting more towards circuitry, it might be more informative to computational models. Certainly we don't want to do multimodal integration, maybe use those EEG frequencies that are associated with feed-forward feedback information. There's more work to be done with 7 Tesla, maybe even with head grading coils to get us to even higher resolution or more flexibility in what we can do. And I think we're still developing the sequences. I think we can get a little bit further in terms of being more specific to capillary effects with our pulse sequences. So with that, I'd just like to end my talk, and I'm looking forward to any questions that you have in the question and answer period in a few days. So thank you very much.